My name's Simon Schultz. I'm the director of the Centre for Neurotechnology at Imperial College London. And I'm going to be talking to you today about neurotechnology for studying mouse models of neurodegenerative diseases. Okay, optical neurotechnology for studying mouse models of neurodegenerative diseases. So in my talk today, I'm going to tell you about a few different technological developments that we've been working on over the last few years. One of them is the two-photon targeted robotic patch clamp system that we've developed, which allows you to use a two-photon microscope to select targeted neurons in the brain to record neural activity using the patch clamp technique automatically. The second technology I'll be talking about is the use of calcium imaging using a two-photon microscope during behavior, so that we can use it to assess how mice perform sort of spatial exploration and memory tasks, and we can use it to probe mouse models of neurodegenerative diseases and how the brain circuitry in those disease states deteriorates. And then finally, I'm going to talk to you about ex vivo two-photon imaging, where we can take the brains of the mice after those experiments and then get a lot more information from them. So firstly, how is information processing by neural circuits affected by neurodegenerative diseases? As you quite probably know, one of the main features of Alzheimer's disease is the development of these amyloid plaques. You can see on this image of hippocampus here, these little bright spots, and they're basically the sort of formation of these clusters of amyloid protein. So that's the hippocampus. You can see the circuitry here. One of the big questions, of course, is how does that amyloid protein disrupt, affect the information processing operations carried out by this circuitry? There's been quite a bit of work on this over the years, but actually one of the things which really brought it to our attention was a series of papers on what's called the aberrant excitability hypothesis. So there's basically evidence that's been emerging over the last few years, in particular from the work of Mark Orrell-Bush using two-photon microscopy recently, that in Alzheimer's disease, some cells, some neurons start firing at higher than normal firing rates, so they're hyper-excitable, whereas other neurons will fire at lower firing rates than normal, so they're hypo-excitable. So we can see this this image here. This is of a small area of tissue, and we're looking over here at basically calcium fluorescence time series. Each of these calcium transients corresponds to sort of firing of one or multiple action potentials. And this is sort of the normal behavior you can see up here. You can see down here, this is a mouse model of Alzheimer's disease. And some of these cells, like the ones in reds, are actually firing more than you would expect that they should be, and some of them are firing less. So there's clearly some disruption of the circuit going on here. And we're really interested in sort of following that through and seeing how that's actually sort of changing what's going on in the circuit. Now, the main tool we use for this is the two-photon microscope, and this basically stems from a basic bit of physics, the physics of fluorescence. So you can see over on the left here, little energy diagram corresponding to single photon fluorescence. So this is sort of what you might see in a typical confocal microscope used in biology. You've got a fluorophore, sort of a sample. Some light is sort of shone on it. It causes an electron to go up in energy level, and then that electron sort of relaxes back down to the ground state, giving off a photon. So there's a photon being absorbed and a photon given off. The photon given off is necessarily of slightly lower energy and therefore slightly longer wavelength. Now, with two photon excitation, you actually have this initial event, which results in the electron going up the energy level, corresponds to the absorption of a pair of photons. The emitted one is basically just the same. And that actually changes the physics a little bit. Because of sort of the physics of this process, in the end, we can see in this diagram here, you end up with this very small point spread function. Okay, so the light that you see given off here is confined to a very small location in space in comparison to this one above here, which is a sort of single photon fluorescence scenario. So because of that, you've got this very small point spread function. You know where the light came from, and you can use that to, for instance, scan that point around in tissue and see what was happening in that tissue. So this is a sort of an example here of, this is a piece of brain, in fact, we're looking at in a mouse. We're exciting our light from a laser. So one of the catches of two-photon microscopy is you actually need a lot of light to have a good chance of this happening. So you have to basically send pulses of high energy light in. So that's coming in here. It's causing this two-photon fluorescence. The light comes back up through the objective lens and then is picked off and detected in photomultiplier tubes. And that's what we see. Now, we can then use this for a number of things. So for instance, if we have a transgenic mouse which has got particular cell classes expressing fluorescent proteins, we can use it to look at the structure of the brain. And here's an example. Here, there's actually 4 small regions that we've imaged in a mouse in vivo. So these are taken in a live mouse. It's a transgenic mouse which is expressing green fluorescent protein in layer 5 pyramidal cells, and that's what we're looking at here. We can see about 800 microns deep into the brain with this sort of fairly standard two-photon microscope in this scenario. So this lets us look at the structure of the brain circuitry, and that's interesting. And we may see changes in the structure corresponding to the development of neurodegenerative disease, for instance, in which case this is a useful method to look at it, but maybe we need a little bit more. So how do we get functional measurements? So one thing we can do is we can use electrophysiology to obtain functional measurements. And in this case, we're still using our two-photon microscope, but we're using it to guide and target a micropipette to do electrical recordings. This work was motivated by some work from Ed Boyden's group at MIT a few years ago, where they developed a robotic patch clamp system. So basically it would introduce a pipette into the brain, bring it down, monitor the impedance at the tip of the pipette, change the internal pressure of the pipette, and when it came across a cell, you can detect that because of the change in the impedance, and then you can suck onto the side of cell and obtain a patch clamp intracellular recording from the cell. But that's all well and good, but that only lets you look at, for instance, you're most likely to see large superficial, most likely to detect large cells near the surface of the cortex if you take that approach. If you're interested in some other cell classes like interneurons, which are sort of more rarely found, for instance, this is not going to be a good way to go. Whereas a two-photon microscope, what you can do is you can take a transgenic mouse that has been bred in order to express a particular type of fluorescent proteins in a cell class, and then you can use that to target the recording. So that was our aim here. This is a system we've developed to do that. So it's basically, an extended version of the robotic patch clamp, which brings a two-photon microscope into the mix as the eyes of the robot, if you like. And what you will do is you will scan around in 3 dimensions and select your targets. So firstly, you would bring the pipette, you would put a pipette in, bring it down to the surface of the brain, detect the tip location. You then scan in 3 dimensions, find the targets that you want, select them, and the robot will calculate the trajectory to bring the pipettes down to those targets. Now, the problem is that as you're bringing the pipette through the brain, it moves the brain tissue itself, okay, through elastic deformation, etc., and that moves the targets. So you would end up at the wrong location if you just went down in open loop mode like that. So instead, you do something like a sort of a guided missile sort of control algorithm, and you're monitoring the locations of the targets as you're going down, and use them to adjust the trajectory on the fly. And then this is what's happening here. We can see over here, we're bringing our pipette down. This is our target down here. We actually image a stack of sort of slices with a 2-photon microscope around the location of the target. We use basically optic flow algorithm to track the movement of the target. We have what's called a contrast autofocus score to look at shifts in the Z direction. And we can see what's happening to the target and then adjust the pipette as it goes down. And our ethos here is really to make as many adjustments as we can as far away from the target as we can, so that when we get down to the target, we're then close to it. And then when we're near the vicinity of the target, we can then go on to an automatic approach mode, and we can just let the blind autopilot algorithm effectively sort of deal with that. And this works quite nicely. So you can see a few examples here of some of the recordings we've obtained in this method. So here in red, this is our pipette. This is the cell that we targeted. So we could have targeted any of these cells. We targeted this one. After we've obtained the recording, that's been filled with red dye. So this image is acquired afterwards. And then we do some of the very sort of standard electrophysiological characterizations of the properties of those cells here. So you can see this is one of these cells here. This is actually a GAD67 containing inhibitory interneuron in the cortex. And here over on the right, you can see another 3 of these sort of same type of cells that we've recorded. It's not limited to that type of cell, and it's not even limited to neurons. You actually can see an example here of astrocytes. So instead here, we've filled our pipette with green dye, and we've used a red dye sulfurrhodamine to label the astrocytes as targets, and we've come down and done a patch count recording onto this astrocyte. And now astrocytes, of course, are not electrically active. They don't fire action potentials. So the electrophysiological characterization sort of shows that over on the left here. Okay, so that's one way of getting functional measurements using the 2-photon microscope. It's actually just using it to guide another recording means. But of course, we can use microscopy itself to obtain functional measurements when we bring in things like activity-dependent fluorescent dyes or proteins. Okay, so now what we're going to be looking at is calcium-dependent fluorescent proteins. So particularly the genetically encodable calcium indicators, GCAM6 is a sort of the most famous example of this. And one of the maybe interesting things about this for us is being able to do this in an awake behaving mouse, which is actually doing a task. So this is our sort of paradigm here. We're actually imaging neural activity during a memory task. Let's look over on the left here. This is our sort of microscope setup. Basically, we have a craniotomy and a sort of window into the brain over the hippocampus. Now, the hippocampus is a deep structure, so using a 2-photon microscope, we can't image it directly from the surface. So we actually do have to aspirate a little bit of the overlying cortex here, and we basically put this cover slip down the bottom. We build this window which lets us look into the hippocampus. And that's chronically implanted. The mice can have that window in for up to several months. Now, in this example, what we are doing here is we're using a green genetically encodable calcium indicator, GCAM6S, and that's shown in the green channel you can see in this image here. At the same time, we use a red indicator as well. And in this particular case, that's M-Ruby. We've actually used a virus developed by Tobias Rose, which will induce expression of both GCAM6 and M-Ruby in these cells. Now, the reason it's useful to use 2 indicators is because with the development of the modern genetically encodable calcium indicators, the baseline fluorescence is actually quite low. So basically, you can only see the cells when they're firing. And that's fine, but if you want to do things like correcting for movement artifacts, etc., you need a more stable signal, really. So that's one main reason we put a red channel indicator in. And that just basically is a sort of a non-functional indicator which sort of shows up in the cell bodies. You can see over on the left here, a movie I'll play here, and this is it's being played at 3 times the actual speed of the image acquisition. And you can see in the green channel here, these flashes corresponding to calcium transients, which in turn correspond to action potential firing by the individual cells. Now, as I mentioned, our main motivation for doing this is looking at it in the context, and you're looking at what's changing in mouse models of neurodegenerative diseases. These mice have amyloid plaques in their brains. So we really want to know, where those amyloid plaques are. And, for instance, if we see changes in particular neurons, are they the neurons that are near the plaques or further away from the plaques, etc. So we can do that, and we can see these images here. You see that the magenta channel here. This is showing methoxy-04 labeling of amyloid plaques. So we can inject into the intraperitoneally into the mice methoxy-04, and it labels these plaques. We then we actually move our laser wavelength down to 720 nanometers to take those measurements. We use more like 920 nanometers for the calcium imaging, and then what we do really over the first few days that we're imaging on any given mouse is we map the brain and look for the locations of the amyloid, look for good cell recording locations, etc. Before I move on to the next slide, I should mention the people who've been involved in this particular, Dr. Marianne Go, who's been really the lead scientist on this, Dr. Jake Rogers, who has, who really basically helped us develop the mouse behavior aspects of this, and Siegfried Prado, a PhD student, and Katie Davey, a collaborator, who basically worked on a lot of the analysis methodology for this. So, as I said, that we actually do this we image during behavior. So how do we do that? The mice actually have to be head fixed under the microscope, the way that we're doing it. So actually this was basically a system developed by a company called Neurota in Finland, called the mobile home cage. And so the mouse is head fixed. There is a carbon fiber cage that it can actually run around on, and that cage is air levitated. It's on top of an airbed to which air pressure is supplied, so that it becomes near frictionless, and then the mice can run around on that, exploring it. But in actual fact, it's the cage which is moving. The mouse itself is staying in one place, but this lets us study some sort of slightly more realistic behavioral tasks than would be possible in, say, in a pure virtual reality scenario. And this gives you just an illustration of some of the behavior that we're observing. So this is our microscope over here. You can see the mouse head fixed under the microscope running around. In this case, he's running around this circular linear track, which is a task we often use. But we can have other variants as well. We can have linear tracks where there's a stop, so it's got to go back and forth. We can have a Y maze, for instance. We've used a Y maze task to develop spatial working memory task as well. And we can have a fully open field as well. The mice just explore all over the field. So they learn to do this over the course of 1 to 2 weeks, basically. They're quite good at learning this circular linear track, so they'll learn that over sort of 10 days or so. We use basically the percentage of time that they're running as a sort of an indicator of how good their behavior is. We want them to be moving around. The reason we want them to be moving around is we want to map place cells. So there are these cells in area C1 of the hippocampus that respond when an animal is in a particular place, and this gives us a sort of a readout of spatial memory. So in order to study those, we need the animal to be moving around enough of the time. Now we can actually also do an open field behavioral task, as we'll see there. In that case, the animal is really just exploring this sort of open field environment. And again, they learn to do that over some period of time. Just to explain these sort of histograms of running speed, particularly in the open field case, it's actually, I would say, probably a slightly longer tail distribution of running speeds than in a, well, the thing we've compared it with is a tethered mouse from David Dupre's group. So the average speeds are about the same, but the distribution is maybe slightly more elongated. Because the kinematics, of course, are not exactly the same as a real-world scenario. The other thing that's different, of course, is that the mice are head fixed, so they don't have vestibular information. And that, of course, turns out to be important. We'll discuss that in a minute. So we can image activity. So here you can see over on the left a region, there's the red and the green channel we're seeing together there. The green channel is obviously of the functional channel, and we basically pull out regions of interest corresponding to individual cells here. You can see there's sort of a few hundred have been pulled out over here. Then sort of down below, so for each of these locations, we've got a line going across here, which is the fluorescence time series. You can see these little sort of lines here. These correspond to the position of the mouse. So the mouse is running around and around this sort of circular track. This is the position of the mouse as it does so. So we can sort of relate these two things. Now this is a continuous signal. What we typically do for the analysis depends on the exact analysis we're doing, but often what we do is we detect the events. So that basically there are sort of discrete events corresponding to calcium transients, and we've detected those events. Now we don't think that those events normally correspond to single action potential firing, the sort of groups of action potentials together. So we detect those events, but we keep the amplitude of the events. And this is what I'm showing up here. We're basically showing all of the events over a period of quite a few minutes of running around the track. And in this case, the size of the dot corresponds to the amplitude of that event. So we find, in this case, reliable place cell activity in the circular track. So you can see here, there are quite a few of these sort of cells. We're showing these examples here, firing when the animal is a particular place in the track and not in other places. You can see overall here, this is basically taking each of the cells. So this is 289 cells. Each of them is sensitive to a particular location in space around that track, and then we're just ordering those cells by the location in track. So you can see we're sort of tiling this space quite nicely. This is over 6 fields of view from 3 mice. We're putting them all together there. And you can look at other indicators as well. What size the place fields are, what amount of spatial information there is, using this approximation of mutual information, which is commonly used in the hippocampal imaging community, or the hippocampal community, to look at this. And we see these numbers, around sort of 0.1 bits per second upwards. Okay, so that was the circular track. Now, I mentioned the open field environment, and we do see some place cells in the open field, but they tend to be much impoverished, is probably fair to say. Very, very low sort of spatial information. And the reason for this is clear if you think about it. Basically you need vestibular information to really properly sort of track position and form place fields in this 2D environment. So this sort of confirms that although there's some spatial information left there, you don't have the level of sort of place cell information that you do in a non-head fixed mouse. It's actually quite interesting that in this sort of circular track, which is much more spatially confined, apparently you don't seem to need that vestibular information to the same extent. So you still get good place cells in that kind of environment. One thing we thought was interesting to look at is just the basic statistics of these cells. So the place cells not all of the cells are place cells. Actually, if you look back here, we've said 289 cells sort of fit our sort of characteristics for place cells out of 721 that were imaged. So here's the place cells and the non-place cells. The place cells tend to fire at more events per second. So here I'm just talking about these calcium transient events, the number of them. They don't necessarily correspond to spikes, so there may be multiple spikes. And the place cells fire more of them than do the non-place cells. Okay, so what about the amplitude? If we include the amplitude of those events, and we can do that here. And what I'm doing here is I'm just sort of integrating the amplitudes of the calcium transient events that occur over a particular time period. So we're giving those in units of delta F on F per second, and plotting those for all cells on a histogram here. You can see I'm doing this on a log axis, and the reason for that is because in the end the distributions end up looking quite log normal, which is actually what we expect from some of the electrophysiology literature as well. So place cells do tend to have higher firing rates if you consider the amplitudes of the events than non-place cells from our imaging. What about the variability? We can look at that. So the mouse is running around and around this sort of circular track. Each time it passes a particular location in space, we can sort of collect the activity at that location. So we get a list of numbers corresponding to activity from that location. We can take the mean of those numbers and the variance of those numbers and plot them against each other for each cell. Okay, so this is what I'm plotting here. There's just two examples here. We'll look at the red one. That's a place cell. Now, as the animal moves to different points in the track, there's going to be a different mean, okay? The mean will be highest at the point where it's actually going through its place field, and it will be low at other places. And then they form basically a straight line in log-log coordinates. The reason for that is that the variance is normally proportional to the mean to some power, okay? And if you take the logs of these, then the slope of this on log-log coordinates gives you this parameter beta, and this beta parameter really sort of characterizes the scaling of the variability for that cell. So we can see a place cell and a non-place cell here. They're not particularly very different in these two examples, but if you look overall over the many hundreds of cells that we've looked at, you can see that actually the non-place cells have a higher variability exponent than the place cells, and that's quite interesting. So the place cells fire at higher rates. I mean, normally we would expect, the higher the firing rate, the more variable, the variance being proportional to the mean. In actual fact, it's the other way around here. The variability exponent there is actually higher for the non-place cells. So although they typically fire at slightly lower rates, they're actually more variable. So there is a fundamental difference here. And one reason this is interesting is that actually, if you go back to the aberrant excitability hypothesis, this may be a very interesting thing to be looking at in terms of a signature of circuit changes due to dementia. We know that some cells are showing this hypoexcitability, some cells are showing hyperexcitability. There's also work showing that there is hypersynchrony in some cases as well. Actually, this variability exponent may give us an interesting sort of signature of what's going on to those cells, and we can relate it to things like amyloid properties, etc. The amount of amyloid in the vicinity. These place cells, if we look at the place cells, they remap. If we take the animal from one environment and put it in another, so you saw this figure I gave before, where we've taken all of the cells and we've ranked them in their location in space, sort of sorted it there and showing this distribution here. You take that animal, you put it in another environment that it's also been trained on, okay, but you keep the ordering the same, then it just looks scrambled. On the other hand, if you resort, as we're doing over here in this environment B, then actually there are place fields. They're just different place fields. So what's happened is the animal has recalled this other environment, and it's actually recalled a whole new set of spatial maps, and a whole new spatial map with a whole new set of place fields. And sort of you can see that here. Now, again, this may be a very interesting sort of indicator of what's going on in dementia. So something for us, a tool for us to use to look at this. The final thing on this is, and really it's just an image at this point, is we can do longitudinal imaging here. We can go back to the same cells day after day. It's somewhat challenging because, of course, at any given time, a different set of cells may be firing. But we can sort of register the locations from one day to the next, and we've been able to do that over up to 2 weeks or so, as you can see here. So again, this gives us the ability to track changes over potentially extended periods of time. So okay, just to summarize the first and the main part of the talk there, with the in vivo imaging, we've developed a pipeline for imaging calcium signals in populations of neurons during behavior. We applied this to spatial memory, to a circular track and an open field task. We're able to take that further. We're looking at a working memory task at the moment as well, where we actually get the animals to go left or right, depending on what they did previously. And we can study these things as a function of things like the amount of amyloid in the vicinity. So at the end of these experiments, we can take the mice and then actually we kill them, we take the brain out, and we can do ex vivo imaging to try and get a lot more information out of that brain. To do that, we use serial 2-photon tomography. Here is a serial 2-photon tomography system. It's called the TissueSite 1000. And the way this works is, so you fix the tissue, you embed it, you image 50 or 100 microns worth using the 2-photon microscope. You then slice that off, image the next one, etc., and then put all these back together. You stitch the images back together to make a 3D image of the entire mouse brain. It usually takes, between 2 and maybe even 5 or so terabytes for a whole mouse brain, depending on the resolution. And then you've got a whole load of slices sort of floating in the bath. Actually, one small advance that we made on this is to introduce a cooling coil into the bath so that the slices actually sink, and that sort of helps improve the image quality. So just to sort of show you an example of doing this with a 5X FAD mouse, so it's a mouse model of Alzheimer's. What you'll see here, the purple you'll see is amyloid stained with methoxy-04, and the gray is an autofluorescence that we use then for registering the brain to the Allen mouse brain database. So you can see the little fly through here where we're showing this. So that's one brain, but what we want to do is actually we take each brain and then we register them to the Allen mouse brain, which is effectively an average of over 1,000 brains, so that then this gives us the ability to start to sort of compile quantitative statistics across many mice. So this is done using the elastics registration algorithm, and you can see the sort of the general process here. Then, sort of having done that, we can do things like automated cell counting. So this is an example. This is a SOX14 GFP mouse. So SOX14 is labeling inhibitory interneurons in the thalamus, and particularly in the dorsal LGN. The visual area of the thalamus, you see a lot of these, and you see some of these cells in the ventrobasal thalamus as well. So to do the cell counting there, this is uses a sort of machine learning approach. We've developed a custom trained UNet deep learning model to do this. And this then allows us to, to compile quantitative statistics across mice and sort of start to look at, what cells are in different areas, etc. But we can use it for more. We can use it to actually trace circuits as well. So in this case, what we've done is we've injected a retro AAV into the infralimbic cortex, and we can then sort of look at connectivity. So potentially we might be seeing connectivity changes as dementia progresses. So this is a good tool for looking at that. So this is at one slice through a whole mouse brain, because we've got all of them for 3 dimensions. I can just sort of zoom in just to sort of show you the level of resolution that we get there. We get 0.5 micron spatial resolution, 5 micron Z resolution, limited by the point spread function of the 2-photon microscope. We can use the rabies virus technique to sort of do that in a slightly more targeted way, where what we're doing is we're using an inactivated version of the rabies virus, but we've basically used another virus in the first place, a helper virus, to put in the thing that will enable the rabies virus to then transmit across one synapses just into a particular cell class. So that, in this case, is labeling inhibitory interneurons in the LGN. We then inject the virus into the LGN. Just those cells then end up transmitting the GFP, which is carried by the rabies virus, back one synapse to other areas. So what this lets us do is to look at all of the cells in other brain areas which are talking to the particular cells that we're actually targeting. In this case, all of the cells that are synapsing onto the inhibitory interneurons in the LGN. And we can then start to trace those connections. So you can see an example here where, so this is the LGN here. This is actually visual cortex, and we'll see that we've actually managed to trace here. I'm showing 2 examples here. We actually start to trace the axons and the morphology of these cells. And we can start to sort of quantify this. This is still, I would say, semi-automated at the moment. So there's a sort of a human in the loop guiding an automated algorithm. But it's enabling us to get much better information about changes in brain connectivity, during conditions such as aging and neurodegenerative disorders. One catch with this method is, so in this case, for instance, the rabies virus infects not just the DLGN, but the VLGN and the IGL, and that's just because of our genetic target in that case wasn't quite as specific as we would ideally have desired. As the molecular technology sort of improves here, then we'll be able to actually sort of add more specificity to these and trace much more precise sort of connections. So to summarize, I've shown you a 2-photon targeted patching robot. It's got approximately human level performance. We then looked at calcium imaging, where we can look at the same cells over weeks and ask questions about how stable the representations in the brain over time, and translational questions such as how effective an Alzheimer's therapy will be at restoring functional properties of circuits. We're doing a lot of ongoing work on the data analysis technology for this. And that's letting us look at things like tracing brain-wide connectivity patterns following in vivo experiments using our ex vivo imaging technology. So I'd like to finish by acknowledging many of the people involved. I've acknowledged Anne, who's a major part of this work already. I'd like to mention here Gerald Moore, who's a PhD student joint with Stephen Brickley's group at Imperial, who did the ex vivo imaging work. And I'd also like to mention our funders, and particularly Mrs. Anne Uren, who basically gave us a philanthropic donation, which really got this work off the ground. So